Bismillah ar-Rahman ar-Rahim. Dear fourth year students, I am Assistant Prof. Mahdi Arbai. Our lecture today is Genetics and Orthodontics. In order to study genetics and orthodontics, we should remember that malocclusion is a manifestation of combined result of genetic and environmental interaction on development of orofacial region. Why do we study the genetics as orthodontists? The first benefit is for orthodontic diagnosis. Being orthodontist, it is essential to study the genetics, hence to consider the genetic role in diagnosis of orthodontic problem, and to understand the cause of existing problem. This may have an influence on the final outcomes of ortho treatment also. The second benefit is to get a promising treatment outcomes. As a general rule, malocclusion with a genetic cause are thought to be less amenable and less treatable to ortho treatment than those with environmental causes. In other words, greater the genetic component, worse the prognosis for successful outcome by means of orthodontics intervention. Hence, knowing the relative influence of genetic and environmental factors would greatly enhance the clinician ability to treat the malocclusion successfully and making the treatment results more promising. We should know some basic terminology in order to proceed in this subject. So what is the meaning of genetics? Genetics is a branch of biology concerned with rules and material bases. Rules that mean that like Mendelian rule. Material bases, we should know that there are some DNA, RNA, chromosomes, alleles, and so forth. For the formation of heredity, characteristics and their inheritance. The mean of genes is the segment on DNA, deoxyribonucleic acid, encoding hereditary information, which is stored in gene and necessary to code the formation of proteins, which are the most necessary to determine the characteristic feature of organism. So the genes are smallest structural functional unit of inheritance, or the most basic and functional unit of heredity. A gene is segment on a DNA molecule that contain all information required for synthesis of proteins that are the polypeptide chain or the RNA molecule. Each gene has a specific position or locus on the chromosome map. We proceed in the next slides to cover this subject. Autosomes. All humans normally have 22 homologous pairs. Each pair comprises one from mother, paternal, and the second from the father, paternal chromosomes that are called autosomes. In addition, one pair of 6 chromosomes is present. Females have 2 long X chromosomes, while the male have 1 long X and 1 shorter Y chromosome. They are different in length and having 50 to 250 million base pairs in each. Alleles are genes at the same locus on a pair of homologous chromosomes. Then one allele is carried out from each parent. These alleles are responsible for variation and uniqueness of an individual. When both members of pair of alleles are identical, the individual is homozygous for that locus and referred to like the texture of the texture characteristics of this individual, like T capital or T small. And if the two alleles are of certain individual or certain locus are different, then the individual is heterozygous for that locus and referred to like small t capital T. Genome is the entire genetic content of set of chromosomes present within a cell of an organism, or it is a genetic mapping of an organism having a genetic information contained in DNA. In 2003, it was found that the human genome is made up of a double helix of deoxyribonucleic acid comprising more than 3 billion linearly arranged chemical nucleotide base pairs named adenine, thymine, cytosine, and guanine bases. Base A was found to pair with T in the DNA double helical structure, while cytosine is pairing with the guanine. This genetic information is normally organized in chromosomes. A chromosome with continuous double helical DNA is wrapped around in the nucleus with a basic protein called histones. Karyotype. This is the laboratory photographic image to look for number and structural abnormality if there is any defect in the number or structure of the chromosome complement of an individual. The genotype and its difference from phenotype. The genotype is a set of genes of an individual that does not change in individual lifetime in general. So it is like a genetic constitution of an individual and may refer to specific gene, locus, or to loci in general. This also can be pronounced as loci or loci or loci. So the genotype refers to the genetic code of the individual. This is all the information that is found inside the individual cell. It is different from phenotype, being that phenotype is an expression of genotype that is visible to other people and can be observed. Phenotype is the sum of all the observable physical characteristics of an individual. The characteristics of an individual have developed as a result of the interaction of individuals' genotype, epigenetic factors, or environmental factors in or around the individual over a period of time. Why we say in the individual? Because if the individual gets a disease, this disease may affect the physical characteristics of the individual or change the development of this individual, especially in early developing time of the life. Or around, that is, the individual may affected by the climate, atmosphere, expression to some stress or habits. So these factors include diet, diseases, drugs, smoking, stress, impaired nasal respiration, or tongue dysfunction. The individual's phenotype is not only morphological but also psychological and physiological characteristics and properties. Epigenetic factors are these environmental factors which have the ability to result in the same genes being expressed differently in different organisms. Therefore, the change in DNA will not be in the linear sequence or structure of the DNA, but in the expression and the properties of gene products of the proteins. For example, obesity is determined genetically but can be expressed differently in different individuals. When some individuals have diet control regimes, express obesity differently from those people who have uncontrolled diet regimes. How is genetic implicated in malocclusion? For orthodontic therapy, morphological characteristics such as skull structure, jaw length and volume, and also sagittal jaw relationship, anteroposterior jaw relationship, are of particular interest. The aim of ortho treatment is to exert a therapeutic influence on the phenotype being treated. The greater the genetic proportion of the anomaly, the more difficult it is to achieve the therapeutic influence, whereas the more environmental factors are involved in the anomaly, the easier it is to treat it. The severity of the phenotype depends not only on the presence of genetics, epigenetic, and environmental factors, but also on the interaction between them over time. How is malocclusion inherited? It can be inherited as a trait. The trait is a particular aspect of or characteristic of the phenotype. For example, number of teeth, arch length, and arch width. The trait can be considered to be of three types: either monogenic trait that develop because of the influence of a single gene locus. It is either autosomal dominant type, autosomal recessive type, or X-linked trait, sex-linked trait, where a trait present on X chromosome is responsible for disease. Male are almost affected, but female can be occasionally affected. For example, hemophilia is a disease of sex-linked or X-linked recessive disease that is present predominantly in male, but female could be occasionally affected or affected and die at early time. The second trait is polygenic trait that are result from complex interaction of multiple genes. And the last one is multifactorial trait that result from interaction of multiple genes as well as environmental factors. So the last one could be more treatable than the first two. How can a trait or such as malocclusion be transmitted? Malocclusion can be transmitted by one of the following modes, either repetitively that occur in from one generation to next generation. So it could be present in the progenitors and then within the current family and in the next generation also. Secondly, it could be transmitted discontinuously. That means it transmitted in the family but not from one generation to other generation, but appear in certain generation and after several generations it reappear. The third one is variable traits, the occurrence of variable expression of different but selected type of malocclusion within several generations of the same family. Penetrance. It is the ability of gene to express a phenotype. The incomplete penetrant gene shows an expected phenotype in a proportion of individuals, in some people, but not in other. Whereas, where gene is completely penetrant, it is always expressed. Expressivity. It is the severity of gene expression, which is not the same in the same individual and in comparison to other individuals. So the extent of severity of gene expression is not the same in individuals, but in varying degrees. So affected person in a single family may show varying degree of disease severity. For example, the cleft lip and palate, it could be present in one person in the family as simple cleft of the lip. In other person may present in as a cleft lip and palate, or could be very simple, which is cleft uvula. Another example is osteogenesis imperfecta, shows varying degrees of severity in different persons within the same family, extending from multiple fractures only or combined with dentinogenesis imperfecta, and in most severe cases it may be combined with hearing loss. How can we estimate the heritability of certain disease. Theoretically, a heritability of a trait can be estimated as 1, a grade 1, if no environmental influence was implicated, whereas a trait with a heritability of half or zero point 0.5 if environmental factor influenced half of its variability, and the other half is influenced by the genetic factors. Again, the heritability of 5 could be more treatable than 1. Methods of studying heritability of malocclusion. The best evidence in establishing the relative contribution of genes and environment in the development of malocclusion can be offered by the following studies. Monozygotic twins. The identical twins develop from single fertilized egg. They are identical in genetic makeup and sex. That's to say, genotype is identical. Then their difference is only in contribution of the environment in their development. The dizygotic twins develop from two separately fertilized eggs at the same time. Dizygotic twins share only 50% of their total gene map or gene complement. Then the difference between them are due to both environmental and genetic makeup. The other studies are parent-child, siblings, pairs, and familial pedigree study. We will define the familial pedigree study or familial studies that is constructing a family tree to study the nature of heritability of a trait, in which the males are denoted as a square, while females are denoted as circle. And by noting who in the family has the trait and who does not have. And also they are the normal female is denoted by a certain color, the deceased by other, the carrier is have to have. OMIM, the current information, all current information, the international or the global information of the genetic or genetic mapping of a human being or maybe other animals can be gathered together in OMIM. So the current information about a human gene and genetic phenotypes can be found in the online Mendelian inheritance in man database, which is created by the Johns Hopkins University and can be accessed free of charge via the National Library of Medicine in the United States. So it is like the main resource in the world about the genetics. For each abnormality in OMIM, the relevant OMIM entries are specified for the relevant genes in these databases and can be accessed at any time because it is free. So, for example, if we are looking for the genetic basis of the cleidocranial dysostosis, which is in OMIM numbered as 119620, we will find that the genes responsible about the cleidocranial dysostosis by 6p21.1. This means that gene 6 is responsible about this abnormality, and which part of gene? It is the short arm of gene, which is called the P arm. Which band of the gene? The band 21 and locus 1 of band 21. The other example is mandibulo-cranial dysplasia, which is result from a defect in gene number 1, long arm, band 22. And the last example is the tooth agenesis, which is also found due to a defect in the gene 22, long arm of gene 22, gene 2, long arm, and band 35. Genome-wide association study. This is again a type of worldwide study. This is an approach used in genetic research to associate specific genetic variation with particular disease. The method involves scanning the genomes from many different diseased people and looking for genetic markers that can be used to predict the presence of disease. Single nucleotide polymorphism, frequently called SNPs, are the most common type of genetic variation among people. Each SNP represents a difference in single DNA building block nucleotide. For example, a SNP may replace the nucleotide cytosine with the nucleotide thymine in a certain stretch of appear in this figure. So the gene cytosine is replaced by thymine in certain stretch of DNA molecule. Heritability of malocclusion. Several studies have shown that skeletal malocclusions are more influenced by genetic, whereas dental malocclusions are more often due to environmental factors. This is because the genetic estimate of skeletal anomaly are higher than those of dental, such as tooth position and number and sizes of teeth. Variation over various family and twin studies indicate that class 2 div1 and class 2 div2 malocclusion are multifactorial interaction of both genetic and environmental factors, while class 3 malocclusion is heavily influenced by genetics. So it is well known that class 3 malocclusion is much more difficult to be treated orthodontically. Heritability of class 2 div1 malocclusion. Class 2 div1 malocclusion appear to have polygenic and multifactorial inheritance. The mandible is smaller than in class 1 and overall mandibular length reduced. Higher correlation between the parents and their immediate family, which means their grandparents and grandchildren and siblings, than in unrelated siblings is noted. However, environmental factors such as tongue pressure, digit sucking habit can also contribute to class 2 div1 malocclusion. Class 2 div2 is more difficult and more related to genetics. Class 2 div2 malocclusion exhibits high genetic influence and is often considered as a genetic trait. Familial occurrence has been evidenced in twin and triplet studies and in family pedigrees. Class 3 malocclusion is also more genetically based. Class 3 malocclusion with mandibular prognathism often genetically runs in families. The most famous example of a genetic trait in humans passing through several generations is probably the pedigree of the so-called Habsburg. This was the famous mandibular prognathism demonstrated by several generations of Hungarian-Austrian Empire. However, environmental factors such as a large tonsil, nasal blockage, premature loss of the permanent molars, and trauma can also cause class 3 malocclusion, but mainly genetic factors are more responsible. Heritability of dental anomalies. Twin studies have shown that tooth crown dimension are strongly determined by heredity. The hypodontia of third molar, second premolar, and lateral incisor shows familial tendency, and it is and fit polygenic model of inheritance. While the heritability of tooth number, the supernumerary teeth most frequently seen on premaxillary region, also appear to be genetically determined. For example, mesodense are more commonly present in parents and siblings. The missing maxillary lateral incisor, which is next to third molars, often exhibit familial occurrence also. Abnormal tooth shape. Abnormality in the lateral incisor region varies from pig shape to microdontia to missing teeth, all of which have familial trends. Female are more predominantly and the association with the other dental anomalies such as missing teeth, ectopic canine transposition, and canine transposition, suggesting a polygenic etiology. The cusp of Carabelli trait appear to be strongly influenced by genes. Ectopic maxillary canine, which is most often palatally malposed canine, is have an inherited trait, being one of the anomalies in a complex and genetically related dental disturbances, often occurring in combination with missing teeth, microdontia, supernumerary teeth, and other ectopically positioned teeth, while submerged primary molars, commonly submerged teeth, especially in mandibular arch, also are genetically determined. The soft tissue inheritance, soft tissue morphology, and behavior have a genetic component. They have significant influence on the dentoalveolar morphology. For example, in class 2, div 1, a short upper lip and high lip level with a flaccid lip tone will favor proclination of upper incisor. It is thought that the external matrix, that means lip and cheeks, are strongly genetically determined, while the internal matrix, which is the tongue posture and behavior, can be influenced by both genetic and environmental factors. There is a theory based by Butler, which is called Butler's field theory in the heredity of malocclusion. Butler suggested that mammalian dentition can be divided into several developmental fields named molar premolar field, canine field, incisor field. Within each developmental field, there is a key tooth which is more developmentally stable, and on either side of this key tooth, the remaining teeth within the field become progressively less stable. For example, within molar premolar field, maximum variability will be seen for the third molar size, which could be microdont, and this is mostly seen in upper third molar, or in its shape that may be not resembling any of the teeth. We know that the ideal form of the upper third molar, for example, from occlusal aspect, is heart shape. However, the upper third molar sometimes coming very small and is not in ideal form or size. Moreover, the root of the third molar, whether upper or lower third molar, sometimes coming short or sometimes long, sometimes fused or separated, and even the separated roots should not be two only. For example, in lower third molar, sometimes lower third molar coming with 3 roots or 4 roots. So this variation is indicating the variation in the size, shape, and roots of the last tooth in the molar premolar field in the Butler's field theory, or in Butler opinion. The second example in the canine field, that is the canine sometimes coming in the maxillary arch, especially as impacted or ectopically erupted, palatally or buccally erupted canine. The last example is the incisor field. In incisor field, the variation is coming in the lateral incisor more than the central incisor, that comes as a pig-shaped lateral incisor or congenitally missing lateral. I would like to thank you very much for listening and have a nice day.